Kei akanui, kei akau rahi tēnā koutou. Ko ai au, no Awherika ki te tonga o ku mātua, I tipu aki a hau ki Johannesburg, kei manakau hau e noho ana, he manakura ki Waipapa Taumatarau I te Māturanga Hawara, he Pākehā a hau, ko Warwick Bag toko ingoa, tēnā koutou, tēnā koutou, tēnā tātou katoa. Welcome to everybody. My name is Warwick Bag, and as I said, I'm the Dean of the Faculty of Medical and Health Sciences. This is Professor Simon Swift's inaugural lecture, which we are very excited to be at, Simon. This is one that I have really been looking forward to for a long time, ever since you first came and met with me last year to discuss your professorial application. I was deeply impressed by the fantastic work that you have undertaken and led over a very long period of time, and so this professorial promotion is thoroughly deserved. For those of you who are not familiar with the process, in order to become a professor, it requires excellence and leadership in research, teaching, and what we term citizenship or service. And Simon has displayed all of those in bucket loads. And so it's a great privilege to welcome Simon to the professoriate tonight. Simon will be formally introduced by a colleague from the Faculty of Science, Professor Frederik van Holsbeek. So welcome to you. Simon will then give his lecture, and then Cherie Blinkron will provide some concluding comments, and then I will have a short talk before we finish up, and then share some time together at the back to congratulate Simon on his fine achievement. But for now, a formal introduction from Frederik. Thank you.
Kia koutou katoa, ko Frederik van Holsbeek toku ingoa. I'm from Science, across the road, quite a bit further down. And formal introduction, I'm not sure it goes very well with my style, but let's try. And I think becoming a professor is really the recognition of the excellence of Simon. His work, his research work has been about providing new understanding of antimicrobial biofilms, extracellular vesicles, microbial communication, and microbial detection. So all about microbes, you know, tiny stuff. He has published 184 papers. That's a lot. That's way more than what you need to become a professor. He could have probably applied 10 years ago. And 2 patents as well. His citation is enormous. He has like 25,000 citations or something like that. Again, above average. And he came here in 2001 to start his lab, and he got more than 31 million in funding, which is also way above average. He has supervised way more students than average as well, like 36 masters and 21 PhD students. And he will talk about his research. So I thought that rather than keep talking about Simon, I will talk a little bit about me, okay? Sorry, I'm stealing a little bit of your time here, Simon. So I met Simon in 2005, so 19 and a half years ago. I know exactly because I was pregnant with my son who just turned 19. I was a very young lecturer. I was pregnant. I looked very young, so young that he thought I was the student and my student was the supervisor. But he was ready to try crazy ideas, especially to talk to a physicist to use light to detect bacteria. And that's the start of our working relationship. And Simon has been very supportive with me as a young lecturer. And a few years later, we wrote a grant together. He taught me English and 101 and biology 101, and I think I'm going into 102 now. I think so. And so it was really a privilege to do some research with Simon and to co-supervise students with Simon, because I think he's the best, amongst the best colleagues I have, and many of you probably share my opinion about Simon. He's very supportive. I think I said that before, not just with his colleagues, but also with his students. You might have seen that they even wear T-shirts. Like, they made the T-shirt to show allegiance. And yeah, keen to try a crazy idea, to give people opportunities, is quite rigorous in his science, always ask the right question, you know, put back the thing into focus, the whole research into focus. And as I said, he told me a lot about biology and a lot about writing. I might have taught him some physics and some French, as you can tell, right? So he's also quite ready to stand up when it's right, when he thinks it's right, and he will support you. So we definitely had hard time working together and good time, and he's always been there. So I definitely owe him a lot, and I'm sure many other people do as well. It's really actually fun to work with him, and he's really good at bringing people together, as you can tell. So I have done enough stealing his time, so now the floor is yours to talk about yourself, Simon. Thank you.
Well, it's nice to see so many people here, so many colleagues and friends. It's always the worry is that nobody will turn up, I think, and that you share a Chardonnay and a prawn straw with the Dean. Not that that would be, not that that would be unpleasant, of course, but so great to see you. Anyway, tēnā koutou katoa, ko ingarangi te whakapakarangi mai ko Sheffield te whenua tupu. Ko Tamaki Makarau te Kainga, ko Simon Swift taho, tēnā koutou, tēnā tātou katoa. Okay, so that's the tongue garbling bit done. So, as I said, as my vowel sounds might have told you, and my accent, I'm from the north of England, especially if you watch Coronation Street or Emmerdale Farm, you'll have become accustomed to my language. I'm from Sheffield, which is I've forgotten how to. This was on right. Don't press that button. Sheffield's just there, South Yorkshire. Famed for its steel industry and obviously its wonderful football teams. And now I call, and for the last 25 years, call Auckland home. And very pleased to do so. So, let's see. Why the title? So, Joe and I managed to sneak a little cycling holiday in between conferences that I was going to a couple of years ago, and and the first hotel we stayed in, we walked downstairs for to collect the bicycles and have some breakfast, and the reception lady looked up and exclaimed, "Ooh la la, la blah blah blah," and I've always thought. Yeah, that was a very pleasant response to my arrival, and I've always remembered it because normally the French receptionist just says non, and so I preferred that one. So I want to deal with some weighty questions today. So. Why is my beard blue? Obviously, because it matches my eyes, but also, why do I keep it blue? You know, it's been like this for a good 10 years, I think. And so the first question is that it's not always blue. It can be other colours. It's just that I think blue's slightly better, and it's the reaction that I get from other people when I was an unbearded fat white man, bald fat white man, should I say, that people didn't really talk to me. You know, students were maybe a little scared of me or a little... But with a beard, people talked to me. So a story that I like to tell is that I was walking down to Science for a meeting there, and a man came up to me, walking in the opposite direction, and just said, "How do you know you're a pirate?" And I was a bit surprised. I said, I don't know. And he said, "Just know you are." And then carried on walking. And I just think, you know, if I can get that sort of response from people, if people are that comfortable to talk to me, then I keep the beard. It also does a lot of hard work at conferences. So I'm a shy, retiring sort, and don't like going out and actually talking to someone. I prefer to stand in the corner and people come to me, and I find the beard is really good for that. So, in inaugural lectures, I've seen in the past many of them, the educational journey is something we talk about. So I'll give you mine. How did I get to Auckland? I started off at Athelston School in Sheffield, my primary school, named after the first King of England who won a battle nearby, I think. I don't have any baby pictures or pictures of me going to primary school in my shorts and things. I just couldn't find any. There are plenty of embarrassing photos to come, so like this one. So, my secondary school, equivalent of grammar school in Sheffield, was Brook. I was obviously a very studious student, and it was quite a long time ago. It was late 70s, early 80s. The UK was ruled by a divisive right-wing government that enriched its supporters and didn't really care for the working people, especially for the working people in northern industrial cities. And my father, who worked at a steel plant, was made redundant because of the redundancies and the cuts that this government made. But and looking back with great admiration on what my father did, he set up his own business. It's still running. It has makes a lot of money now, and my brother runs it very well. So something good came out of Thatcher, but not a lot. So it's also important at this stage to show and associate yourself with famous alumni from your school. And I've seen various, sportsmen and great scientists come up. Brook was more the arts. We had the year I came, Sean Bean left, but he's our probably only famous student, I think. And my music teacher was this guy who's Tom Bailey, who after 2 years of teaching me music, or not teaching me music, decided that he was going to be a pop star, and actually succeeded, which to everyone's amazement. And he's one of the people in the Thompson Twins, if you don't recognise him and weren't around in the 80s. He's still around. I think he's doing a concert in New Zealand in the near future. And both Sean Bean and Tom Bailey have, I think, strong connections with New Zealand, which is interesting. Anyway. Brook School was closed in 1989, demolished, and a housing estate was built on it. My brother now has a house on there. I left Brook after my O-levels, that's NCA 1, and went to Richmond College. I was looking for, I think, a more adult experience, and to be treated more like an adult when I was being taught. Richmond was a great college, a very diverse group of students, from many overseas students, and a really good attitude to more relational teaching and giving people the responsibility to take responsibility for their own learning. One of the highlights of being at Richmond College was, and doing A-levels in biology, was the biology field trip, which was a few days camping and doing ecology at Bempton Cliffs on the Yorkshire coast, where we looked for puffins. I think it's one of the only places on mainland UK where the puffin breeds, and the gannet colonies, and of course the rare dotterel. That pub has also now closed and has been converted into a house. So things change, but I had very good memories of Richmond College. I left in 1984. That was another year of social upheaval in the UK. We had the miners' strike, where Arthur Scargill and the miners took on the Conservative government and struck for quite a long time, eventually losing, and lots of people lost their jobs as that industry was almost dismantled. The photos there are from the Battle of Orgreave, where police and miners clashed, and Arthur Scargill was arrested. And Orgreave was a mile from my house, so it's a very local. Anyway, so there were challenging times to go to university, and I'm grateful to my parents for supporting my decision to do so, and also helping financially. Some things don't change. Richmond College closed in 1997, and it was demolished, and a housing estate was built on top of it. That is the last time I'm going to say that. 1984, I went to Nottingham University, and I spent a bit of time making the decision of where to go to. I thought, it was interviewed at a number of places, and I think I also interviewed them. And Sutton Bonington, where the School of Agriculture, where I went, was just outside of Nottingham. And it's a fantastic place, fantastic environment. I really enjoyed my time there. And I studied applied biochemistry because I think because of the economic times, I wanted to do an applied degree, something that maybe would lead into a job. Obviously, it didn't. But, so, this is my year. It was the 80s, so mustard and white were trendy colours. I graduated in 97, and this is one of our iterations of our football team, our School of Agricultural Sciences football team. Really put there to remind me to say, we had a great timetable system. Wednesday afternoons were sport. So if you wanted to play sport, there were no lectures, you could. If you wanted to do anything else, you could. We didn't need gen ed courses set for us that we had to pay for. Anyway. I think it's a better system. And I'm going to digress, because at that time, what I call my teaching philosophy was really the core of it was pretty set. I wasn't that keen on school and being told what to learn and things. And I really enjoyed my time at Richmond, where we were given a lot more freedom to actually take responsibility for our learning. And while my first 2 years at university were a lot of fact learning, which I wasn't 100%, I was good at, but I really loved third year, where we actually read things. And our dean, the first thing he said to us when we came in all fresh-faced was, "You are reading for a degree," and not listening to videos on Canvas for a degree, which I think is something that we've maybe lost. Anyway, so I'm not that keen on lectures. I try not to lecture when I can. And the Twicker is the joke book of Sheffield University. And this definition of a lecture, the process of transferring what is written on the lecturer's page to the student's page without any of the information passing through either of their brains, is something that I think sometimes resonates with me. I'm sure colleagues will disagree. But there is pressure, I think, on whether we should do these big transmissive lectures. Students don't seem to like them, they don't turn up, often because they're working to pay their fees, and educationalists are questioning their value as well. So I think sometimes in the future, we may need to think just how we deliver the knowledge to the students. And generative AI is here. It's not going to go away. We have to embrace it. We can't just punish students for using it. So my teaching philosophy is really teaching's what I do, and it facilitates. But the important thing is the learning, and that's what the student does, and we need to really remember that when we design new courses and think about how we deliver our teaching sessions and how we assess students. Opinionated diversion over. Or that one anyway. So I do think the inaugural lectures should be a time to give your opinion as well. However, much people might disagree with it. Anyway, I stayed at Nottingham because I liked it, to do my PhD with Gordon Stewart on tryptophan synthase in E. coli and the interaction between the alpha and beta subunits. Very molecular. I've never had an interested audience talking about that, so I won't talk about it again. After my PhD, I got a postdoc in Auckland and came to work with the late Ray Ralph and learned a lot both about myself and about how to be a scientist in a new place. And I'm indebted to Ray for that opportunity and his time and his mentorship. I also did a lot of sightseeing and ran a couple of marathons as well, so it's a very enjoyable time, I think, of my life. I then went back to Nottingham as a research fellow for quite a few years, especially working with Paul Williams at the main site of Nottingham University and then in the Queen's Medical Centre. And you can just see the top of our institute built in there, if you look carefully. And then I was fortunate to be offered a job at the university, but back in Auckland in this faculty in 2001, and I've been here ever since. It's a great place to work. I really enjoy it. I really like the people here. I think it was a good decision for me. And over the years, I've done worked on many different things that I'm not really going to talk about because I don't have 4 hours. I've worked with lots of great people, great students, great colleagues, and had lots of fun. So this isn't the end. There's about halfway. But I thought, how do I talk about things that I've done, but only talk about a couple of things? And so I'm going to. I asked myself a couple of questions that I thought I might ask someone else who was giving an inaugural lecture. The first is, what was my first scientific paper? The first paper that I was an author on, that, I suppose, put me on the permanent record of planet Earth as a being here, which was important to me. And at the end of the 80s, PCR, polymerase chain reaction, certainly the scientists will have heard of it, was invented and just becoming popular. And we got the first PCR machine in the University of Nottingham. It was very exciting. We had lots of people from other labs come to use it, and we used it to amplify some genes and clone them and to look at how they worked. And we showed that they were biochemically functional, which hadn't been done before. And this is, looking back, this was the first paper to actually publish something that was probably very obvious. But we said in that paper, at present, much work with PCR is directed towards development and application of gene probes. But if the application process results in error-free replication of the original template, then the potential for cloning and expression of genes using PCR is prodigious. And I think that's been borne out. But yeah, many people in here will have done it, and I bet none of you have cited the seminal work. Anyway, my most exciting result. I really like science. That's why I've never got a job. I've always sort of been a scientist. And I get a kick out of it. I really enjoyed, doing experiments, designing experiments, and analysing the results, interpreting the results, and talking about the results. And so I thought, when was I most excited? And obviously it's an early thing. And it was just after I'd done my PhD, and before I came out to New Zealand, I had 3 months, and this is when we did, or maybe 6 months, this is when we did most of this work. And it starts with Barry Bycroft and Paul Williams in the Department of Pharmaceutical Sciences in the main site at Nottingham. And they were really interested in the carbapenem antibiotic, really important antibiotic then, really important now. And they wanted to understand its biosynthesis so they could obtain intermediates, so that they could do chemistry on it to make better semi-synthetic carbapenems like had been done with the penicillins. And they envisaged this hypothetical carbapenem biosynthesis pathway that would synthesize that was present in bacteria to study. And they hypothesized that if they took a mutant bank, they'd make mutations in the biosynthetic genes, and those mutants wouldn't be able to make the antibiotic, and they'd be able to screen for those. And then they'd be able to cross-streak them to look for cross-feeding, and that any late pathway mutants might cross-feed early pathway mutations, and they'd identify stable biosynthetic intermediates in the pathway. And that all worked fantastically until they identified the molecule, which was this molecule we call OHHL. And that's not an intermediate in the carbapenem pathway, no matter how you move the bonds around. And so they were a bit perplexed. It had already been patented as well, so they were perplexed and pissed. And it had been described earlier as the auto-inducer of a phenomenon that we're probably all aware of now, bioluminescence. Quite esoteric, not thought of as, in the microbiology fields as the main thing you should be working on. And at high cell density, this molecule, the OHHL, accumulated and activated gene expression in the bacteria, and all the bacteria start to glow, and you could see them. No point them doing it at low cell density, because you wouldn't see them, there wouldn't be enough bacteria making light. And we thought, well, maybe this is how the bacteria is regulating carbapenem production. It doesn't make it early in growth phase because it doesn't need it, but if it's colonized a niche, maybe at high cell density, it needs to produce an antibiotic to protect what it's got. And if Erwinia and Vibrio, which are quite different bacteria, do it, then what about other bacteria? Is it a global phenomenon that we've missed? Could it be an opportunity? And it became this phenomenon called quorum sensing that some of you may be aware of. Now, what we did was, first, we made a reporter, so something that could detect, a biological agent that could detect the signal, the OHHL. And how we did that was we took the Lux operon from Vibrio fischeri, deleted the Lux I gene so that this organism couldn't make the signal itself, so it would be dark, but if you provided it externally, that would activate the system and the bacteria would glow. And we used this to screen all the bacteria in the lab. We had a big teaching lab with lots and lots of different species, and we found a set of gram-negative bacteria that all activated this sensor, so very exciting. We then made gene libraries of all of those bacteria and used the reporter to screen those libraries to find the ones that were making this signal so we could find the gene. Now, it was 1991. Some of you won't have been born. If you were an academic in 1991, the most important picture was you looking at with a sequencing gel. Mine has been lost to the annals of time, but otherwise I would have shown it, but there definitely was at least one. And nowadays, we'd send off the DNA and it would come back sequenced very quickly, and we'd take the file and analyse that. I had to take the gel and work up the gel and work out what the code was and write it down and then type that into the computer and find out and then analyse that there. But now, if you were doing that now, you'd go to something called the internet and paste it in, hit go, get the results. Great, easy. There was no World Wide Web. We didn't have this sort of thing. We had to write our own scripts. Not my talent, I must admit. But we had something called SeqNet, which was part of the Janet system which was linked together some UK universities. This is the Daresbury lab in Manchester, or outside of Manchester, where the supercomputer lived. Probably two Amstrads stuck together. And we analysed the DNA using those programs. We identified the open reading frames, got the hypothetical translated proteins, and used those to probe the database that they housed on the computer to find anything that was homologous to these proteins. And the first one I did was one of our teaching strains, an Enterobacter, because the clone from that strain was the smallest and fastest to sequence. And I put the sequence together, I sent it off to the, found the protein, or all the open reading frames, sent those off to the computer, went home, got in early the next morning, and opened the files. And the first one I opened, which had the biggest protein on, had full homology, full length of the protein to the Luxi protein that made the OHHL. So not only were they using the same molecule, they were making it with the same enzyme, which was really exciting at that time. And I was so excited. I saw the name and the homology to the protein, and I pressed print because we didn't have email or anything like that. I pressed print, and then realised that it was quite a large document, and 400 pages of dot matrix printer later, I had a nice pile that I annotated the important piece of information for, took it downstairs to my supervisor's office and left it outside because it was still quite early in the morning and he wasn't in. And then I went into the lab and did things, waited. Now, Gordon was a fantastic supervisor, and he was really enthusiastic about his science and results and things, and he always had at least one bottle of champagne in the fridge for any good, unlike me, any useful results, any good results that he expected were going to happen, and any publications and things. So that was a really good day. We had champagne. I was very popular, and it really, when things aren't going well in science, I think back to that day because it was good, right, and exciting, and this is the eventual putting all the proteins together. There's not a huge amount of homology, but enough to get me excited. And we published in this paper. And quantum sensing's been good to me. Before I came to Auckland, we published quite a lot of papers. We didn't publish anything in the super impactful journals, the Nature or the Science, mainly because we tried, and they sent it back saying, oh, not of any real interest to everyone, not a cutting-edge major discovery. And then certainly in the next 6 months, a former president of the ASM published something on a different bacteria, not Erwinia and carbapenem, but Pseudomonas and virulence factors in Science, and a fellow of the Royal Society published something on agrobacterium in Nature. And that's always sat badly with me. So I don't publish in Nature or Science. It hasn't stopped these papers being highly cited. I think the quality of your research comes through in your citations over time, and if it's good work, people read it and people cite it. So you don't have to publish in Nature. So I'm going to jump 25 years because we need to get to the end. what do I think is important now? And lots of things, but one of the things is biofilms. So at the end of my time at Nottingham, we started working on biofilms and quorum sensing because the biofilm is a high cell density mode of growth. You'd expect it to quorum sense. And this is there's some nice recent pictures that a summer student, Fengaru, took with the help of Jackie Ross and the excellent people in Beirut, who have always been great supporters of the work that I do, and I'm always grateful for such a wonderful resource in this faculty. We can see the bacteria, they're expressing a red fluorescent protein. We can see the biofilm matrix, the polymers that the bacteria secrete, and we can see in the overlay that the bacteria are encased within the matrix. And this is really important in something that I like to call antibiotic failure. So we know about antibiotic resistance. Antibiotic resistance is really important, something that we definitely need to take a lot more seriously than we do, but it kills us in slowly, not quickly like COVID, so we don't do anything about it as quickly as we should. But there's also antibiotic failure. So you don't need to have a resistance gene to not be killed by an antibiotic. And biofilms in an infection, which is most infections, are encased in their matrix. It's difficult for the antibiotic to penetrate them. And when antibiotics do, the cells inside, they're not growing very quickly or at all, and they can tolerate quite high levels of antibiotics. Antibiotics inhibit things and kill bacteria that way. So if the bacteria is not doing much, difficult for the antibiotic to kill it. So we're interested in how we can kill these biofilms. Now, try and do a half-hour talk in half a minute. So we have a set of osteomyelitis isolates that are all methicillin susceptible Staphylococcus aureus. So they're not MRSA, they're not the nasty staph, they're the boring, harmless staph. But they're not. If we test them in the lab, they're sensitive to flucloxacillin, which is a methicillin-type antibiotic. But we don't want to kill the bacteria we grow in the lab. We want to kill the bacteria that are causing the infection in the patient. So this is the biofilm. So in the lab, if you've been in my lab, you'll see we have lots of these bioreactors growing biofilms. And in vitro, at the MIC, at 10 times the MIC, at 100 times the MIC, this antibiotic doesn't touch the bacteria, doesn't kill them, the different isolates, so the symbols on the graph. So you don't need a resistance gene to be insensitive or not be killed by the antibiotic. And in a mouse model, we can make an infection, and that's not killed by the antibiotic either. So, sorry. Here we have a rat, and we've made a little defect in the leg bone, and we've inoculated it with biofilm. So not broth-grown bacteria, because we know that they're sensitive to antibiotics. Biofilm bacteria that are tolerant to antibiotics, and actually the ones that we'll see in real life. So this is important if you're doing this type of work. Inoculate with a biofilm, not with broth-grown cells. And as part of the animal health of this animal, so it doesn't get systemic infections, it's given a big dose of flucloxacillin at the start. That will prevent any disseminated infections, but the biofilm infection isn't touched. And we're able to use this model now to to look at ways in which we can better kill biofilms, better treat them. And one thing that that we've been looking at is something that we have quite a lot of in New Zealand, a milk protein, lactoferrin, and we mix that with flucloxacillin, and we can see we get a much better kill of the bacteria that's causing this biofilm infection in the rat. The challenge now, and that's working with Jill Cornish and Jingwan Wen, the challenge now is to pharmaceutically prepare that, formulate those active ingredients and be able to use them to treat patients. A younger me would make a joke about this. Am I transdisciplinarity is vogue at the moment in the university. Basically, it's me, or the scientist, in this case the microbiologist, with collaborators to form a multidisciplinary team. But that's not good enough now. We also need to have relationships with industry or with communities to really understand what their problems are so that we can target our research to give solutions to those problems, which I think is a good way of going about things. I'm wholly supportive, even though I made a joke about it. And I've done a lot of transdisciplinary work over the years, and it's not just started a few years ago. So we've always been transdisciplinary. Multidisciplinary teams of chemists, engineers, physicists, microbiologists. In the hybrid polymers program, led by Ralph Cooney and the biocide toolbox that followed, we had transdisciplinary projects with FoodSafe, with working with Fred. We had transdisciplinary projects as part now of the New Zealand Food Safety Science Research Centre. Just about all we do is transdisciplinary. It's working with industry to provide safe food for everyone. And now the university is funding transdisciplinary projects. I've had the pleasure of being part of a team led by Joyce Campbell from Fine Arts that's really doing water-centred transdisciplinary engagement with Wairoa communities, looking at how we can look at flooding, look at the microbiological quality of the water, many other questions. So I really, if you haven't done or don't do transdisciplinary research and you are a researcher, I recommend it. Interacting with colleagues from different disciplines, they do loads of cool stuff, much more difficult than what I do, I think. And interacting with industry, you get to understand what the actual problem is, not trying to make it up yourself, not getting distracted by the shiny mechanistic thing over there that only 3 people will ever be interested in. And if you can do research for people that they actually want, then maybe the reputation of scientists also will increase and governments might value us a little bit more. Anyway, so now for the thank you. Now the thank you section is the longest section. I'd like to thank my academic colleagues, my early career mentors, Gordon Stewart, Paul Williams, who gave me so many opportunities, Ray Ralph, who gave me my first postdoc and taught me so much more about being a scientist that I hadn't, and from a different perspective, Stanley Molloy, who I worked with at the University of Illinois in a 6-week sabbatical, who taught me again so much about asking the right question. Colleagues at the University of Auckland, Naresh Singhal, who was the first person to recognise me as someone who'd be a good collaborator in 2002 or something. And we've just got, we've collaborated all the time. And we've just got a grant through to the second round of Marsden, so we're still hopefully going to be working together. Fred, obviously, Jennifer Craig, Anthony Phillips, and Ralph Cooney, and all these people have invited me into their project really because they needed a microbiologist, and hopefully being glad of the experience and provided, I think, an environment for me to be able to develop my projects and things I think are important from within what they're doing. My colleagues from chemistry especially, who in hybrid polymers and in Biocide Toolbox, have done some great work making things for me to test. Normally the conversation goes, how much do you need? I'll say, about a gram. How much you got? Or about 10 nanograms? And we it's like the Moroccan bazaar. We eventually decide how I can use 10 nanograms. But Paul Kilmartin, Yadranka, Margaret, Jan Young, Karnika in engineering, all great people to work with. And now working with Vijay, I'm working with Sue Young as, and their wonderful students is also carrying on that work. And people I work with a lot, Maria, Susie, Jill and Jing, all really good people to work with, great projects, keep me really interested. Joyce for taking me in as part of the transdisciplinary project, and Saeed for collaborating with me or allowing me to join one of his projects with his great student, Indu, who works so hard and very productive science with all these people, and I'm very grateful. And of course, Cherie and her chicken. Now, I was visiting my parents a couple of years ago, and my mum said, Simon, who does the work when you're away? Because I'd been at conferences and things. And I was sort of I was going to sit down and give a considered explanation. My brother came straight in and said, it's the same people who do it when he's there. And yeah, I think he's probably right. And so those people, I've been blessed with fantastic technicians to work with: Benedict, Jared, Adeline, Latifa as a teaching technician, and especially Jan, who these are the people who establish the assay in your lab. These are the people who train the students how to do the work. These are the students who put their arm around the student when it doesn't work and say, oh, it's all right, Simon didn't mean to be mean. Thank you. You're all wonderful. I've not finished. Also, my students. I've got 2 slides of students because I've had so many. My honours and master's students. I think master's students and honours students are great in the lab. They always work really hard, always get good results. And my PhD students, some of these were my master's students and honours students. You might see them just slightly older in this photo. Especially Maria, who was my first primary supervisor, Charge, who's in the top left, who texted me this morning from Canada to wish me all the best for today, which was great. When I went down to that graduation in 2000, whenever, I was heckled by people on the street as, look at Harry Potter. As we've progressed, I now get heckled, look at Henry VIII. But anyway, sign of a life well lived, isn't it? I'd also, and Cherie promised lycra, I think, to some people, so work-life balance is really important. I get most of mine, and have had most of mine, from either running, exercise, riding bicycles, and I've had great fun with lots of people over the years, especially people from university, the Sutton Bonington Sabres. We used to do lots of marathons and half marathons. The lab group who came once to a race and decided it was, decided it was maybe just something for me. The guys I go mountain biking with on a Monday morning. My son Luke, who enables me to buy, or has enabled me in the past to buy lots of bicycles, and because you need at least I think 5, maybe 6 as a minimum. And Jimmy Dalton and Cody Mankalo, who got me running again a few years ago to do Auckland Half Marathon, and I'm always indebted to them for that, because I lost a load of weight and I got fitter, and it was really good. And it's great to have friends like that. And of course, family who put up with me. My mum and dad, who encouraged me at school to do well, and in a positive way. Encouraged me to go to university and supported me and gave me money while I was there. And supported me when I decided to move to the other side of the world, because they understood that it was important for my career. And obviously Joe, for all the happy times that we've had together, and hopefully all the happy times that we'll have in the future. And of course the kids in our blended family, and certainly Luke and Zach and William and Daniel. Not always a pain in the ass, generally. And I think that's it. I'll just leave you with this photo, just to, or collage, just to remind Cherie that she should be succinct.
So I have the pleasure of doing the concluding comments here as head of department, but actually more personally as a colleague, a friend, a collaborator for many, many years. I have to say I apologise for the lycra. That was actually my fault. So like Fred, I actually just wanted to tell you a little bit about our journey, and it really picks up on a lot of the things that Simon's talked about. So our own collaboration actually started with a conversation in early 2011. I've got a very vivid memory of having a conversation with Professor Anthony Phillips in the attic in SBS, wondering if bacteria and human cells can talk to each other during an infection. As a surgeon and a cancer biologist, we realized that we needed a microbiologist on this project. So we needed a go-to guy, a so-called bacteria guru, as I called him in the email, dated to then Dr. Swift, dated the 11th of March 2011, asking for advice about our crazy idea. And Fred also mentioned these crazy ideas. And this was noting before le infamous Le Barbe Bleu. So by the end of 2011, we had 2 summer students, Nick and Sylvie, working on this idea The next year, we had our first honours student submit her thesis, Sasha, and this really cemented a lot of the protocols and the methods that we are still using today. This little idea absolutely snowballed into a big project. I've listed training around 10 postgraduate students, 3 research fellows who are actually all in this room, including myself, multiple technicians, as you've already seen, producing 12 publications on EVs to date, including 2 international guidelines, bringing in a few million dollars in external research funding. It led to the establishment and Simon's leadership of the bacterial EV task force with the International Society for Extracellular Vesicles. He's not good at blowing his own trumpet, is he? A deputy editor role with their flagship journal, and new collaborations that he's pursuing with the data from Jiwon's paper, actually, with new collaborators in China. So EVs, the thing that we work on, are just one small thing, as you've seen in all the work that he does. You've heard about his other passion of biofilms, and there are many, many others. As the bacteria guru, he's forged many, many collaborations with networks across the university, across globally, and all of which have been approached with the curiosity of, and I'm so pleased you said this, a proper science nerd. Ask him about fusiform vesicles, and he will absolutely bend your ear off. And he has a let's give it a go attitude, which Fred also alluded to. And I think that's the sort that you get from having a good northern upbringing. Personally, by experience, I had the same. Professor Swift, so nice to say that, is always willing to pitch in and do the hard yards with his research and his teaching, as you've seen. I've actually often found him in the lab washing out bottles and emptying the bins. He likes to get his hands dirty. But other skills not yet mentioned. The ability to find a craft beer bar in any city in the world, proof of evidence. Fluency in 2-finger touch typing. And more seriously, he's an incredible advocate for students. He provides them with opportunities, with support, but he gives them independence and really promotes their own responsibility, letting their own voices come through. I asked a couple of past PhD students to comment on his supervision. The first said, and we have to guess who this might be, Simon has been an incredible mentor. His wisdom, guidance, and unwavering support were instrumental through my PhD journey. And of course, I'll never forget the Jaeger bomb-fueled pep talks along the way. Who could that be? Another said he was a fantastic supervisor and boss, supportive, understanding, chilled, and with a much-needed sharp sense of humour, as you've seen today. So these statements really sum up the time and encouragement he's given to his students over his career. I nearly fell off my chair when I was counting them up, supporting nearly 60 postgraduate students, which is absolutely incredible. And they've all gone on to various academic and non-academic roles all over the world. So to finish, he'll be pleased that I'm finishing, I would like to close with the actual Professor Swift-approved summary that we actually planned a few weeks ago while drinking beer in Vienna. And it goes, well done, Professor Swift. Hasn't he done well? Let's not keep you any longer from the beverages and nibbles. Thank you very much.
Thank you very much, Sharia. Thank you, Simon, for an outstanding lecture. I think that was an advert for biomedical science that we should put far and wide. That is such incredible work that you have done through your career and the transformation over time. I can only think of you typing out those codes and what a pain in the butt that must have been. And now we just push a button. So thank you for your work. Thank you for your contribution. I wish I could grow a beard like that because I'm an introvert and I really struggle to talk to people. So thank you for sharing your secret sauce. But it's now me between you and the drinks. And one final thing, I'd like you to come forward and just receive a little token of appreciation and remembrance of the evening. We just have to stand for Chris over here to get the picture. Let's give Simon one more round. Thank you. Please all join us for some drinks and nibbles outside. Oh.